Okay, so I was just saying that RNA is another coding molecule similar to DNA, and it's involved in the replication of DNA and in protein synthesis, which is what we're going to look at now. So I was also saying, we're having a few technical problems, but there are 3 different types of RNA. So there's only one type of DNA you need to know, but there are 3 types of RNA here. So we have mRNA, tRNA and rRNA. So if we look at them all individually, they all have different purposes and functions. mRNA stands for messenger RNA. Okay, so it's found in the nucleus and the cytoplasm. It's found in the nucleus because it needs to take a message from the DNA template to the ribosome where the proteins are formed, and it enters the cytoplasm to do that. It carries a coded message from the DNA to the ribosome where it meets with tRNA, transfer RNA. It carries triplets, the triplet codons that we mentioned earlier, in a message which is converted into RNA, from DNA into RNA. So it's still, guanine will obviously be coded into cytosine, so G, it will be a C on RNA. T, thymine, will be an A, adenine. The difference is that where there is an A on the DNA, for adenine, there will be a U instead of a T on RNA. Okay, I hope that makes sense. So uracil instead of thymine. So if it's an A on the DNA strand, where you would expect it to be T with DNA, with RNA it's U for uracil. Hope that makes sense. When we look at tRNA, this stands for transfer RNA. So this is found in the cytoplasm near the ribosome, where proteins are made. tRNA translates the message from mRNA into another coded message, so that the coded message with the tRNA is made up of anticodons. So they're just opposite of what the mRNA has carried, opposite codons. So these anticodons are then used to code for specific amino acids. So each triplet that comes along has given anticodons by tRNA, and then which codes for one specific amino acid. rRNA you don't need to know much about, but it's called ribosomal RNA, and it's found within the ribosome. Surprise, surprise. And it's involved in the functioning of the ribosome and joining the amino acids together to form a protein. So that's just a breakdown of the 3 different types of RNA that you need to know about. So we're basically going to look briefly at how the DNA is copied and coded for by the mRNA, messenger RNA, and eventually translated into another code by the transfer RNA, the anticodons, which give us specific amino acids which are joined together in a chain to make a protein. So the protein is also a polypeptide, remember, because it's made up, poly is Latin for many, peptide is protein, it's made up of amino acids, so it makes up a protein. So I have one diagram which I want to go through with you, this guy here, which basically puts everything together, which I've just said, in a way that's easier to understand. So here is our cell with our nucleus with the DNA in it, and this is a picture showing you how it's all twisted up together, and then before protein synthesis can take place, the DNA helix has to unwind itself. So it unzips itself, and the base pairs, you can see they're still together here. C is with G, C with G again, T is with A, G is with C, A is with T, and then they begin to separate. So before mRNA can be formed, these base pairs along the DNA molecule will separate from each other. So here we have part of the DNA, and along the bottom we have our messenger RNA coming up with a code. So here's the T on the DNA, here's A, here's C with G, A on the DNA, remember, you would expect T, but because it's RNA, it's a U for uracil. G with C and G with C. So this section here is messenger RNA. That's then carried as a triplet codon, so just in three at a time. So you have C, A and U, that's your mRNA, as it says, and that's your triplet codon